Hi, I'm Kevin Yang from UC Berkeley, and welcome to this talk about our paper, Improving Molecular Design by Stochastic Iterative Target Augmentation. This is joint work with Wengang Jin from MIT, Kyle Swanson from the University of Cambridge, and Regina Barzilai and Tommy Yakula from MIT. So the 15-second version of our work is that we propose a data augmentation approach which substantially improves the state of the art in molecular optimization by over 10% in 2 tasks, and is also broadly applicable and useful in other structure generation tasks as well, as we will demonstrate in the program synthesis domain. Okay, so first I'll give the brief rundown of our actual task setup and method for molecular optimization, and then after that I'll go into more details and analysis. So suppose you're trying to make a new drug, say to treat the COVID-19 virus, by stopping the virus from entering human cells. That would be pretty useful. And maybe you've even come up with some promising candidate for your drug, like shown here. But it's just not quite there yet. When you test it, it only stops 78% of the viruses from entering human cells. But you really want it to work on over 90% to effectively treat the infection. So you want to make it more potent, better at stopping the virus. And you don't want to redesign a molecule from scratch. You just want to modify or optimize your existing molecule a bit. Okay, so that's basically the task of molecular optimization, which is the main task we focus on in our work. We frame molecular optimization as essentially a translation task. The goal here is to quote-unquote translate an input molecule, such as the one shown on the left, into a similar output or target molecule with a higher score for some property of interest, in this case the fraction of the virus that it stops from entering human cells. The output isn't necessarily unique, since you'd be happy with any successful modification of your molecule that improves its property score. And then the dataset for molecular optimization is just a collection of these input-output molecule pairs. Okay, let's talk about what makes molecular optimization hard. In the real world, the way we measure most properties we care about, such as the ability to stop COVID-19 from entering human cells, is by doing some kind of actual physical experiment, like a lab assay. The problem is that this can be slow and expensive. And an unfortunate result of this is that datasets in chemistry tend to be much smaller than their counterparts in NLP or vision. A lot of the time, datasets in chemistry are only on the order of hundreds or thousands of examples, as opposed to the hundreds of thousands or millions or more that you might see in NLP or in vision. So that's really one of our key challenges in molecular optimization, that the datasets are often just much smaller than we'd really like. Now, since the key challenge here is that the dataset is small, this motivates trying a data augmentation approach. So our method, stochastic iterative target augmentation, is a data augmentation meta-algorithm that can be added on top of any existing translation architecture for molecular optimization. Let me try to interest you by showing the results first. For molecular optimization, we ran experiments on 2 architectures, vSeq2Seq, which is a sequence-to-sequence model using string representations of molecules, shown here on the left, and HieraGNet, which is a state-of-the-art model using graph-based representations, here on the right. And we use 2 standard molecular optimization datasets from prior work, QED and DRD2 here, and our metric is the fraction of input molecules for which our model samples at least 1 correct output out of 20 tries, measured by a ground truth evaluator. And in each chart, the base architecture is in blue, and our augmentation of that architecture is in red. You can see we substantially improve over the base architecture success rate on both tasks, achieving new state-of-the-art by over 10% in absolute gain on both tasks. And these results also demonstrate that our methods both agnostic to and complementary to improvements in the base model architecture. And actually, our method can be quite useful in other domains as well. Here you can see we get quite strong results in the program synthesis domain. So I'll give the full details of the setup later, but the point here is that our data augmentation approach in red beats not only the baseline in blue, but also a strong reinforcement learning approach in green. Okay, so now that we've seen the results, let me summarize the method in the context of molecular optimization. The first step in our data augmentation procedure is to repeatedly sample a bunch of candidate input-output pairs from the current generative model. So for example, you could generate a bunch of candidates that might be effective against COVID-19, but you don't yet have any guarantee of quality for those candidates. Some of those candidates might be great at stopping COVID-19 from entering human cells, but others, maybe most, wouldn't be. So we wouldn't want to use these as training data directly. Now the question is, how are we going to filter down to the good molecules only, the ones that really do stop over 90% of the COVID-19 viruses from entering human cells? So for each possible drug candidate that we sampled before, we need to check what percentage of the virus is no longer able to enter human cells when in the presence of the drug. This corresponds to the problem of property prediction, shown here. Given a molecule, you want to predict the property score, in this case, the fraction of viruses that it stops. And actually, it's intuitively easier to do property prediction than to generate a molecule from scratch. We can make an analogy to programming here. It can take quite some effort to write a program, depending on the specs, and this is like our constrained generation problem of molecular optimization. On the other hand, it's quite easy in comparison to execute your already written program on some test cases, and just check if the program is correct, which is the analogy of prediction. So for the sake of the analogy, you can almost think of the drug as a sort of biochemical program, and that it's harder to design a new one than to check when an existing one is good. And of course, this program synthesis analogy is very relevant, as we will show experiments in that domain later on. Okay, so returning to our method, stochastic iterative target augmentation, we're going to filter down our sampled outputs using a property predictor. So in the COVID-19 example, you'd use your property predictor to predict which molecules stop more than 90% of viruses from entering human cells, and then only add those molecules to the training set. Finally, we're going to train our model on the augmented training set and repeat. That's the main idea of our method, and I'll go into the full details later on. Okay, so that wraps up the short quote-unquote intro poster session of our work, or poster session intro of our work, and I'm going to spend the rest of the time discussing first some more details of the task setup in the context of real-world molecular optimization, then give a more thorough explanation of our method, show some additional empirical analysis, and then finally show that our method can be broadly useful in other structure generation tasks as well in the program synthesis domain here. So let's begin with the details of the setup. As we discussed before, in real-world molecular optimization, the ground truth property score is generally a lab assay. This poses some major problems in practice, namely that it can be very slow and expensive to evaluate. This also means you cannot afford to do this ground truth evaluation except at final test time, since the turnaround time for designing a molecule, actually physically making it, and then testing that molecule in the lab and getting the results can be on the order of months. So when you run your computational models, you're going to validate them with some fast and cheap in silico, or computational predictor, which you train on a dataset of real ground truth values that you collected before. Since the in silico predictor is just some computational model, you can run it whenever you want. And then our evaluation setup is designed to mimic this real-world setting as much as possible. Since we don't have access to the lab assay, we treat an in silico predictor from prior work as our ground truth, which we're only allowed to use at test time. Then for our cheap predictor that we're allowed to use anytime, we construct a proxy predictor, which is trained on the output values of the pre-existing in silico predictor. And actually, we don't need any data on top of the molecular optimization dataset of input-output pairs that we already had. We just train our proxy predictor on the pre-existing predictor's values for the molecules in our molecular optimization dataset. Since if you think about it, you needed those property values to construct those input-output pairs in the first place for your molecular optimization dataset. Finally, I want to discuss our metric a bit more. As I mentioned before, we define success as any one of 20 translation outputs passing the property score threshold measured by the ground truth, as well as passing the threshold for similarity to the input. This metric makes a lot of sense because it's pretty reasonable to propose a batch of 20 drug candidates to test in the lab in parallel. And you'd be happy if even one of those 20 drug candidates actually worked out in the end. But even with this 1 in 20 definition of success, if you look at this graph of results for the previous state-of-the-art higher GNN model, which I reproduced from before, the blue baseline higher GNN is still far from perfect on both tasks, which just goes to show how difficult molecular optimization really is. Okay, so we've discussed our task setup and evaluation and how it mimics how one would use molecular optimization in practice. Now I'm going to go over our method again with some additional details and observations. All right, so as the name suggests, the core piece of our method, stochastic iterative target augmentation, is the target augmentation step. In our training dataset of input-output pairs, each output is matched to a single correct output. But in molecular optimization, there could be several different correct outputs for a single input. And our goal here is to find those other correct outputs and add them to our training data. Okay, so the first step is to take one of the inputs in your training data and feed it into your generative model a bunch of times to get a bunch of candidate targets, as shown here. But if we use these candidates directly as new training targets, we'll just get vanilla self-training, which we show in our ablations performs really poorly. So how are we going to narrow down these candidates to only the good ones? Well, as we discussed previously in the quick rundown, property prediction is intuitively easier than generation, for the same reason it's easier to test a program than to write it. And also recall that we're allowed to use the proxy predictor whenever we want. We just can't use the ground truth evaluator. So we're going to filter down the candidates to only the good ones which, 1, pass our proxy predictor, and 2, are similar to the input, and the ones that pass both checks get added to the training data in our data augmentation process. And we do this whole procedure for all the inputs in our training set, so we end up finding some new correct targets for every input in the training set. Finally, we take this target augmentation procedure we just described and loop it with our generative model training. So we augment the data, train the model for an epoch on the augmented dataset, then regenerate a new augmented data set with the improved generator, train the generator again for another epoch, and so on. So putting it all together, our method substantially improves over the state-of-the-art in molecular optimization by over 10% on 2 tasks, just reproducing here this results slide that I showed earlier. Okay, and finally, I want to make a couple of additional observations about our method. One, we show in our paper that you can interpret our approach as stochastic EM, where our target augmentation step is the E step, and the model training on the augmented dataset is the M step. So there are some theoretical reasons as well to believe that our method will work. Second, why an iterative approach? The reason to do target augmentation iteratively, where you're doing it after every training epoch, is because at first your generator might be poor, so target augmentation will struggle. But as your generator improves over epochs, target augmentation may be able to discover more and better new correct targets, which further improves the model, and so on. And then finally, since you have the proxy predictor already, you might as well use it at test time too. So what you can do is you can filter the outputs of your model at test time too, using the proxy predictor, when you're proposing your 20 official outputs to submit to the ground truth evaluator. But we show in our paper ablation that the training time target augmentation actually matters much more for performance than this test time filtering. All right, so that was the more detailed version of our method. I want to show a couple of empirical analyses now, which show that our model is in fact doing good things in addition to the previous success metric. First, I'll discuss 2 different metrics other than the success metric that we can use to evaluate our model. The first is this freshet chemnet distance. This is the molecular analog of inception distance in images. It's basically this notion of distributional distance between embeddings using a pre-trained model. So here, we check the distributional distance between the model's final predicted outputs on the test set against a reference set of gold targets. You want to see lower distance to indicate a better distributional match. That's indeed what we see from comparing our augmented model in red to the baseline in blue. So what this shows is that our model is not just overfitting to the success metric that we optimized for, or to the proxy predictor. The second metric I want to show is diversity, which is defined here as the average distance between 2 different correct output modifications for the same input. And you can see that our augmentation has much higher diversity of outputs than the baseline. And then one last interesting result I want to highlight is that our method is actually pretty robust to the proxy predictor's quality. You can see here in this diagram for the DRD2 task that even when the proxy predictor is quite poor compared to the ground truth, our model still does quite well. So in this diagram, the blue line is the baseline, and the red points are the augmented model with varying quality of the proxy predictor. The far left point is the oracle performance if we directly use the ground truth as the proxy predictor, which is cheating, while the second point from the left is what we use in our main paper results. The performance is actually not much different. And actually, if we look at the x-axis, the test set RMSE between the ground truth evaluator and the proxy predictor has to go all the way up to 0.4 on this task, on a property that only ranges from 0 to 1, before you start to see a large drop in performance. So this analysis is indicating that our algorithm is pretty robust to the proxy predictor, so it might be viable for more difficult to predict properties as well. Okay, and you can find the rest of the analysis and ablations in our paper. And finally, I want to move to a completely different domain, program synthesis. The task we use in program synthesis is the Karel dataset. Karel is an educational programming language where you control a robot to move in the grid world. The inputs to our generative model in this task are a collection of input-output test cases, and the output of the model is a program which is supposed to pass all the test cases. Each training example consists of this collection of input-output test cases and 1 correct program, although there could be several correct programs in theory, and at test time we evaluate the output programs on the held-out test cases which weren't fed as input. And adapting our target augmentation method to program synthesis is actually fairly straightforward. So between training epochs, we augment the target set of programs for each set of test cases by having the model sample programs and just check if they actually pass the test cases. So this domain is actually kind of perfect, since we don't even have to learn neural predictor of correctness in this domain. We can just run the program on the test cases that we were given as input and just check if they pass. And the programs that pass get added to our training data for the next epoch. So here are the results again. Our metric is top 1 generalization, which just checks if the first output program is correct based on whether it passes the held out test cases. So we use a baseline architecture from prior work labeled MLE here, and just reproducing the results slide from before, we see that our augmentation in red outperforms not only this baseline in blue, but also the strong reinforcement learning baseline in green. Okay, great. So in summary, we've proposed a meta-algorithm for improving performance in structure generation tasks, where we augment the target set guided by a property predictor. We substantially improved on the state-of-the-art in molecular optimization by over 10% in 2 tasks. We showed that our method is broadly applicable and useful in other domains as well. You made it all the way here. Thanks for watching, and we encourage you to check out our paper for the full details.